Today's presenter is Wayland Rushing, and Wayland is had a long career, and over this career, he's led CMC development programs for a wide array of pharmaceutical products, including parenterals, inhalation drugs, drug-device combinations, and other therapies with complex delivery systems. Dr. Rushing is a subject matter expert in HPLC and GC method development and validation, stability testing, extractables, and leachables, program design, and regulatory submission requirements, and has drafted multiple IND and NDA submissions. Dr. Rushing is a co-author of PDA Tech Report number 65, Technology Transfer and Tech Report number 76, on identification and classification of visible nonconformities in elastomeric components and aluminum seals of parenteral packaging. Following his talk, there will be a chance for questions and answers, which you can type in the question box, and he will then answer them. And I'll now turn it over to Wayland.
Thank you, Eric. I'd like to thank everybody for joining us today. I hope you find the webinar informative and look forward to any questions that you may have at the end. So kind of going over the agenda, kind of what I'm hoping to cover is first is trying to get us on the same page as what are extractables and leachables. They do differ, of course, from normal degradants or impurities. So it's important to understand where they come from and why are they different and how are they similar in some cases. Why do we perform extractables and leachables testing? It's a big driver. Sometimes it catches people by surprise when they get requests from the FDA to perform the testing. So it's important to understand what drives that. How do we determine what are the leachables, which can be tricky depending upon your specific drug product? How do we know that a peak in the end product, is that a leachable? Is it an impurity? Is it a degradant? What's required for leachable methods and how that ties into the overall leachable stability program design. And then just a couple of case studies that kind of wind down the discussions, kind of some examples that we've ran across over the years. So the first thing is to understand what is the difference of what we're talking about here. Of course, a degradant, as defined by the ICH Q3B, is an impurity resulting from a chemical change in the drug substance. So you're talking about something directly related to your drug substance that's in there, and you're expecting it to grow over time, potentially, because it's degrading from your product. When we're talking about extractable leachables, they're really not related to your drug substance whatsoever. So an extractable is a compound which you can, under aggressive laboratory conditions, you can pull out of various different materials. And we'll talk about those materials here shortly, but they're not your drug substance materials. They're more in terms of what the drug substance or drug product come in contact to. Now, a leachable is typically, it's a subset of the extractables, and that under the normal storage conditions actually do migrate from the contact surfaces into your final drug substance or drug product, which then can end up getting dosed to the patient. It's important to understand that extractables don't always equal leachables. Extractables don't always leach, which is a good thing since we're under aggressive conditions. When we do the extractable studies, we're going to see a lot of peaks potentially, a lot of compounds come out. So it's good that they don't all leach, but it's important to understand what is potentially there. But leachables don't always extract. This is a little bit more harder one to conceptually understand. There are some specialized conditions. I have one example of it as we talk, but it's important to understand that it is possible to see a leachable in your final product that you didn't characterize during the extractable studies. So where are they coming from? The first and most obvious is the primary packaging components. This is the final container closure system of where your drug product or drug substance is coming into direct contact with your material. So in this case, that I have up there is a syringe. So in this, what we're looking for is materials that are going to come out of either the barrel, the stopper, the needle that is going to migrate into your drug product and then get dosed to your patient. The second are secondary packaging components, and this is a potential source, especially if you use a polymer final container closure system. So the example I have up here right now is very common in industry. It's a laminated foil pouch that's heat sealed. It's very commonly used for unit dose types of systems that are typically molded in plastic. The issue is that that laminated foil pouch, especially when it's heat sealed, creates volatile compounds, which then goes and can migrate into the materials. The second thing is, from a secondary packaging components, is that we have to worry about labels. So labeling can be applied to primary packaging components. It can be applied to secondary packaging components. The challenge with labels is that, of course, you have a glue and inks. Glues are almost by nature volatile. Inks have several volatile components with them. Unfortunately, when you place those on certain polymer materials, the inks and or the adhesives can migrate very aggressively right through your container closure system into your final drug product. So it's important to understand how the secondary packaging, how the labeling plays into the potential sources of leachables. And then you have your associated or dosing components. These are materials that are used at the time of dosing. It's really in terms of, for example, medicine dropper, IV bags, that type of associated components that are used to administer the dosing. Of course, for the leachable studies, we're primarily going to look at the leachable stability studies, the primary packaging and the secondary packaging. It's important to understand that it can also come from the dosing components, but typically you don't perform stability studies on those. Additional sources which sometimes can come into play, manufacturing components, especially in the area of single-use systems, which are all polymeric. It's important to understand that during the manufacturing process, your drug substance or drug product or the reaction mixtures are coming in contact with various different materials, which can then leach materials into your final product and get dosed with them. These you can think of as manufacturing impurities. We typically characterize those through the manufacturing process and at release of the product or the drug substance. A lot of times you end up having to not monitor for those that are coming from this source on stability because they're just coming in from the manufacturing process. They're not going to grow over time. But it's important to note you can get leachables from the manufacturing process. And the final one for the product is supply chain materials, your cardstock overboxes, and even in extreme cases, some of the shipping materials. One of the largest recalls that has occurred in the drug pharma industry was actually a result of tribromal anisole, trichloroanisole, which essentially leached from the shipping containers through the boxes and into the final product, so that when people were opening up bottles, they would smell musty and dirty, and they wouldn't want to dose them. Now, of course, the leachable stability studies can't really determine this, and this is just an extreme case. What I was using here as an example was there's lots of different sources that potential leachables can occur throughout the entire, starting from the manufacturing chain, your raw materials. It's important to understand them all when you're doing the overall risk assessment. Today, of course, we're going to be focused in on those leachables which are coming from your final container closure system that your drug product or drug substance is going to be stored on long term from the stability standpoint. So why do we do extractable leachables? The first is that it is a regulatory expectation. Extractable leachable data is required, or at least an assessment of extractables and leachables is required for marketing applications. It's also required, of course, to be able to perform a safety evaluation. What is being dosed to the patient from a safety concern? But then also, the secondary driver is to understand your container closure system and how it's interacting with your product so you can gain better product knowledge and potentially reduce down your risks. Because by understanding what potential leachables you may have, because that not only necessarily concerns a safety in terms of what's being dosed to the patient, but in some cases, these leachables have been known to interact or react with drug products, specifically biotech products, and potentially either reduce down the efficacy or cause secondary interactions within the body. So from a regulatory requirement standpoint, this section here is from the 211s basically for the drug substance, drug product on the small molecule side. Then you can see that both of them have very similar wordings, is that basically the contact material shall not be reactive or additive as to alter the safety, identity, strength, and purity. So it's the additive part that we're looking at here, where they're not supposed to add anything that's going to impact the overall quality, i.e. leachables. When you added the 600s or the biological sections, they switched from that and started actually using the terminology of a leachable. So now we see the first things from the regulation that they're calling it a leachable, i.e. materials that can come from the container closure or the contact surfaces and leach into your final product. We do have some regulatory guidances that have been issued by the FDA to help. This is, for example, the 99 guidance on the container closure systems. We have the 93 guidance for nebulizer meter dose inhalers, 98 guidance on MDIs and DPIs. And in 2002, we saw a pair come out for nasal and spray inhalers, and then drug products in semi-permeable container closure systems. The one thing that you'll notice is the majority of these are focused around inhalation products, which was kind of the first area that really got a lot of scrutiny. They're also the highest risk. And in semi-permeable, which makes sense because you're dealing with a very permeable plastic material. But you also notice is that there's really nothing to deal with parenterals, dermals, solid orals, everything else. They're really kind of focused in here. These guidances are very good to tell you that you need to perform extractable leachables, and in some cases why. And again, it really doesn't get into how. One of the most helpful items from the guidances is this risk assessment table to help understand from the FDA's point of view and realistically a safety concern, what's the risk associated with the product of potential leachables. So on the left-hand side, what we're looking at here is the degree of concern or risk associated with the route of administration. And this has to do with how the patient is taking it. So of course, the highest risk are going to be those that are either bypassing the body's natural defenses, i.e. parenterals, or are being dosed directly to an organ that may be susceptible, for example, the lungs. When you're dosing drugs directly to the lungs, there's some safety concerns in relation to that. And of course, the low risk would be oral administration and dermal administrations in some cases. And then across the top is the risk associated with, as they refer to it, component dosage form interaction. And a better way to put it is, what is the risk that you're going to have leachables in your final product? And of course, the highest risk are going to be those solution-based formulations which have a solution interaction with or contact with the materials. Medium and low risk, you get into basically powders or solid orals, capsules and tablets. So by understanding this risk assessment table, you can understand essentially how far you have to dig down into the data sometimes and what you end up having to present. Now, it's important to understand that from this risk assessment table, just because something is rated as low risk does not mean there's no risk. For example, one of the largest recalls for a leachable was actually in relation to a solid oral tablet. So it's important to understand that there is still risk even for a low risk route of administration and a low risk dosage form for a leachable. So just because it falls into that category, you still need to consider what the potential risk is. So since the guidances don't give us a whole lot of information on how to perform extractable leachables, we have to fall on either best practices or now we have the USP chapters that will help us understand and provide some guidance. So the PQRI, or Product Quality Research Initiative, is an organization that's a conglomeration between academia, industry, and regulatory agencies that basically publish best practices or industry standards to execute studies. They did publish the best practice document for inhalation, orally inhaled, and nasal drug products back in 2006. Which is really the first time that putting out there down on paper, here is how to perform extractable leachable studies. It does focus specifically on inhalation products. The USP very recently published their chapters 1663, 1664, and 1664.1. Of course, 1663 deals with specifically extractables, whereas 1664 and .1 both deal with leachables, with 1664.1 really specifically, again, dealing with inhalation products. It is important to note that because of the way that the USP is set up, is that these chapters in the USP are informational. Any chapter that's over 1,000 is informational only in the USP. So they're good guides, they're best practices, but you can deviate from them, but they have really great information in terms of how to execute some of these studies or give some background. So if we look at stability studies, the intent of stability studies and why we do them under normal conditions vary over basically the phase and the drug development process. In the early phase, of course, we're performing stability either to support clinical, i.e. generate the stability study that's going to show that our clinical dosing solutions are good over the clinical study time frame, and then also to help us with the formulation development so we can understand what the true stability is or start gaining that formulation A may give us a better product than formulation B from a stability standpoint. And then we get into phase 3, which we really get into our registration stability support, the stability studies that we're going to end up filing that is going to be based for our shelf life or retest dates. And then after that, when it becomes commercial, i.e. in phase 4, what we're looking at here is really our stability commitments, what we're committing to from a commercial, and then any stability support for life cycle support, for example, altering the dosage form, adding new dosage strengths. Now, if we start to look at how extractable leachables fall into this, traditionally in phase 1, phase 2, early phase studies, very little was done from an extractable leachable standpoint. And as I continue here shortly, you will understand why is the amount of effort that's required to perform extractable leachable studies can be fairly intensive. So again, since things are changing very often in phase 1, phase 2, very little, a lot of time is performed. Typically, what we typically recommend is when you get into phase 2 or toward the end of it, this is where you really need to start doing a lot of assessment for extractable leachables on your final container closure system. Because here is you're making the final choices, what polymers you're going to use, what manufacturing process is being finalized. So by understanding and starting some evaluations early, you can help try to alleviate some of the risk running into your registration. Of course, in phase 3, I have the here with the too late. The issue is that to do a full-blown extractable leachable studies can take a significant amount of time. If you only start after you've started your phase 3 stability studies, because of the length of time sometimes, depending upon the product and the risk you run into, it can really start to impact or jeopardize what your proposed filing date is. Sometimes it can take 6, 12 months or longer to run through the entire extractable leachable study design. So it's important to make sure that you don't wait to the last minute. A lot of places, the reason that they wait to the last minute is because they don't understand extractable leachable studies. They realize that, oh, I guess we have to do this now. We're getting ready to file. We're trying to file in 3 months or 6 months. Unfortunately, for a lot of products, that's too late to go through and do the proper extractable leachable study. So if we look at what a full-blown E&L study looks like, this is a kind of a generic high view over and of an extractable leachable study from beginning to end. You start with the information gathering. What's the contact materials? What's the container closure system? You set your analytical evaluation threshold, the shorthand, the AET, which I'll cover how we set that here shortly. Essentially, that's how low do you have to look down to. You perform your controlled extraction studies. If you're going to control your extractable leachables at your incoming material, you will set or develop and validate control methods for your extractable container closure system. You may perform, then you'll move into performing a leachable analysis. And this is really, for this leachable analysis, it's really trying to say what potential leachables are there, identify those leachables, and then develop and validate specific leachable methods for your final product. These are the methods that are going to move into your release and stability testing if you need it. And then, of course, you're going to be performing leachable tox evaluations on anything that you see that gets above a certain level. Now, it's important to understand that there is somewhat of a feedback loop sometimes. You're sometimes on stability, you're going to identify a new leachable, which may then you have to go back and see whether or not your current method is appropriately validated for that leachable, or after you've identified it, you need to do some additional bridging validation. This is why sometimes it can take, starting in phase 3 or after you've started your phase 3 studies, unfortunately, a little bit too not enough time to get through this. So this gives ranges. Of course, 1 to 2 months. This is the amount of time to gather information, do the evaluation. Perform the controlled extraction studies can take 3 to 6 months, depending upon the number of container closure systems that you have to evaluate and how complex the systems can end up being. If you don't have any materials available, you may want to perform a simulation study so that you can see what your potential leachables are before you move into your development and validation of methods, and then finally your stability. So as I said, it can easily take 6 months, 12 months to walk through the process to the point that you're able to start generating data on your stability. Needless to say, having to try to generate enough stability data for your registration filing. So again, the point I'm trying to drive home here is don't wait too late before you start your extractable leachables evaluations. So now, how low to go? This is a very interesting and discussion topic. So for degradants, the ICH goes through in their guidances and basically defines levels that they're going to have to you report down to identify and qualify. 0.05%, 0.1%. However, those don't apply to leachables. The ICH actually has terminology in it that says this does not apply to leachables or impurities which derive from the container closure or contact surfaces. So it's also important to understand that there's no regulatory guidance that specifically says here is what you need to go down to. Now, there are some things that we can use as guides. The first is the genotox guidance that's out there. It lists you a threshold, or a TTC, toxicological threshold level of 1.5 micrograms per day. However, in that guidance as well, it has a caveat that specifically says this guidance does not apply to leachables. It says that you can use, the framework of that guidance to evaluate it, but the specific 1.5 micrograms per day does not directly apply to leachables. The one area from a regulatory standpoint that we do end up with some guidance is from the ophthalmics division. Now, they have stated in a couple of different presentations that they give, and the reference you'll see down there, is what they consider to be required for ophthalmics leachables. And this is a 1 ppm reporting threshold, a 10 ppm identification threshold, and 20 ppm qualification. And that's still the basic levels that they expect when filing for ophthalmics. So for all the other ones, there's nothing else out there. Now, if we go to the PQRI, from the Product Quality Initiative, they instituted this safety concern threshold. It is essentially the level at which that if you have any compound or leachable that's below this level, there's a negligible risk that there's going to be a toxicological or a toxicity issue related to that compound. And it's based upon looking at the high-risk leachables, their toxicity, and really what are the bad players that we've observed over the years. And this is presented as a total daily intake, similar to the genotox guidance. Now, for inhalation products, the PQRI recommends a 0.15 micrograms per day, so fairly low level. Now, the current thinking of the working group from PQRI with the PODP, which is the parenteral ophthalmic division, is 1.5 micrograms per day for parenterals. Now, again, this only applies to unknowns because once you've identified a leachable, you can perform an individual risk assessment for that. Excuse me. So as we do the evaluations, it's important to note that really right now we have inhalation recommendations, we have parenteral recommendations, and everything else, there's really nothing out there. So it gets handled on a case-by-case basis, and the FDA handles it on a case-by-case basis. Looking at the industry when you're filing to make justifications for what safety concern thresholds that you are using for your overall evaluation. And if anybody doesn't know the reference that I have up there for the dose makes the poison, it's actually a quote from Paracelsus. And the short answer is that everything is poisonous. It just depends upon how much that you end up giving or getting into your body. So it really is how much is getting dosed to the patient on whether something is poisonous to that patient or not. So how we calculate the analytic evaluation threshold. So we have the safety concern threshold, but that only applies to how much the patient is getting. Now we need to convert that over to analytically, how low do we have to go? So here you can see the overall equations kind of for converting between the safety concern threshold into that AET. So to do that, we need to understand in that container closure system, how many doses are there in a day, how many doses are in the device, and then using those ratios and those amounts, we're able to generate basically what that equivalent micrograms per device is. And then from that, we can then convert that into, if we're doing extractables testing, we can convert that into a micrograms per gram by looking at the AET in terms of the component weight, or we can convert that into micrograms per mil for the final drug product solution, if we're talking about, for example, a parenteral solution, by using what the overall fill volume is. Now, here's an example of why dosing can be so important. So the example that I'm presenting here is, let's assume a parenteral product, two different parenteral products, and they have the exact same container closure system, a glass vial with a elastomeric rubber stopper. They have the exact same fill volume, so scenario 1 and scenario 2. Same fill volumes. The patient's going to get 1 dose per day. The only difference is that in scenario 1, the patient's going to get the full 5 ml dose. In scenario 2, there's 20 doses per that vial. Now, historically, if you're using it, a lot of people would say, well, I've done extractable leachables for scenario 1, so that's going to apply to scenario 2 because it's the same glass stopper, same vial, so there's the same risk. So I can apply what I did here over to the other one. Unfortunately, when you run through the math, what you'll see is that's not necessarily the case. So we have the 1.5 micrograms per day safety concern threshold, the same number of doses, but the doses of vials, of course, here are different. They have the same fill. So what you'll see is that if we were to do it for scenario 1, yes, we could apply it to scenario 2 because the AET we have is significantly lower. But let's say we did the opposite. We did the extractable leachable studies for scenario 2, but now we're 20 times higher than we needed to be for scenario 1. So we couldn't use the information from scenario 2. So it's always important when starting these evaluations to make sure that you take into account the dosing information. So when we look at the two different products, the pharmaceuticals versus biologics, there's some differences and similarities here for being able to set up the studies. First, for pharmaceuticals, i.e. small molecules, you're going to end up with potentially a fairly wide range of container closure systems. Everything from solid oral dosages to parenterals to MDIs, dry powder inhalers, a very simple to very complex systems, which can make the extractables portion and identifying what your potential leachables are difficult. The solvent characteristics are not impacted by the active ingredient. So because of they're typically a fairly, even for high concentrated ones, the solution that it's dissolved in or the contact surfaces really are what drives the solubility or the leachable profile, not the API. The API doesn't really impact the solubility profile of the final product as much. And primary concern for a small molecule is really what's the toxicity of the leachable. When we move over to biologics, things change. First is we're dealing with, more than often than not, very simple container closure systems, vial stopper, syringes, things that are, you end up with 3 or 4 contact surfaces, nothing really that complex for the most part. However, there are a lot of significant differences. The first is that the solvent characteristics can be heavily impacted by the biologic. So what this is referring to is if you have an aqueous system, but you have a biologic, a protein or an antibody or something like that in there from a biological standpoint, you can actually have leachables migrate into the final product that you typically would not expect to be soluble in an aqueous environment. That's because those leachables are associating directly with the biologic. You can think of it as the biologic itself as being kind of a micro solubility environment. So you end up with these leachables coming out of the container closure system and then basically associating directly with the biological product. So you can be surprised by what you can see for biologics from a leachable profile of things that you would not expect. And there's actually two different areas of primary concern. The first is the toxicity of the leachable, and this is, again, what the final patient is. However, with biologics, we also have to worry about what is the reactivity of the leachable with the biologic. There have been numerous cases to where a leachable has caused deamidation, glycosylation, aggregation, and a variety of other issues with biological products. Unfortunately, there's no real good way to predict when a leachable may or may not react with a biologic. So a lot of these are after the fact when we've noticed it. But it is important, sometimes you can minimize the risk by starting the studies earlier to understand if there is going to be potential for that type of interaction. So of course, everything starts with, after you've gathered the information, actually doing the extractable studies. You can't just jump into leachables because you don't have an idea of what the full range is of what your potential leachables are. So the first thing is to understand what can possibly come out of my container closure system or the contact surfaces. And this is done by performing extractable studies. Typically, it's done by generating an asymptotic extraction with multiple different solvents. And the reason why multiple different solvents, not just, for example, your drug product, is that you want to generate some worst case scenarios to ensure that your extraction studies are covering what can leach into your product, especially over long periods of time. So the solvents that you choose should cover the polarity and pH range of your final drug product. If you have a non-aqueous, let's say that you have a ethanol-based drug product, well, you want to make sure that you choose two solvents that are higher in polarity range. One higher, one lower, and then one typically in the middle that is very similar to the polarity of your drug product. If you're dealing with a completely aqueous one, you may also have to look at higher and lower pH. For example, if you have a drug product that needs to be at pH 6, but can be like 6 to, let's say, plus or minus 0.5, it is very, very important to understand that the leachable profile can be significantly impacted sometimes by small changes in the pH range. So when you do the extraction, sometimes it's important to also not only vary polarity, but vary the pH. And then you're going to go in there with vigorous or aggressive extraction techniques. There's a variety that you can use: oxalate, reflux, sonication, microwave. In the end, they all give very similar extraction profiles. They have some pros and cons to them. But with whatever extraction technique you use, you don't want to get too aggressive. And what I mean by too aggressive is that you don't want to physically break down the material that you're extracting. Because if you start to do chemical or physical changes to that material during the extraction process, well, then you're going to be artificially creating extractables that's going to complicate what you need to look for to be able to tell what you need to monitor for in your leachable study. So things to look out for there is that when you're extracting a polymer, that it's changing colors or breaking down or swelling. Those are all very good signs that something chemically is likely occurring, and it may impact what your extractable profile is. You're then going to analyze those extracts by a variety of analytical techniques. HPLC, looking for potentially semi-volatiles, non-volatiles. Again, this is typically using HPLC mass spec. Using GC mass spec, looking for volatile compounds. ICP for metals. And I'll talk a little bit here about for 232, 233, and ICH Q3D compliance. How do we do that from extractable leachables, and can we build that into being compliant with AICH or USP? And then depending upon what your contact surfaces are, if you have rubber material, elastomeric material, you may need to worry about special case impurities, polyaromatic hydrocarbons, nitrosamines, turmeric-captobenzothioazole. These are all special case high tox compounds, which used to be very prevalent in elastomeric materials. Now, the good thing is that most manufacturers have gone to some fairly good lengths to eliminate them, but it's important to understand that in some cases you may have to be concerned with those. So from a leachable development standpoint, there's a couple of ways to move forward. So we've done the extractable studies and we see 100 peaks. which in some cases is not exaggerating. You can see 100 or more peaks in some cases. Now, a couple of paths forward is you could develop methods to look for leachables using all 100s, or you can use a couple of different routes. The first is you already have some aged product that has been stored in that container closure system, either from your phase 2 studies, if your container closure system hasn't changed, and it's the same formulation, it's the same container closure system. You can use those phase 2 studies, for example, if you have a 12-month or 24-month samples, excuse me, that you can go in and look at those age samples to see are there leachables present there using the same methods that you use during your extractables profile. This allows for a very focused leachable method development. You're using those aged products to help guide you into it, into specifically looking for what you need to identify and what method you need to develop. And it really can shorten that development time. Of course, that is not always available because sometimes you make container closure changes before you move into phase 3. You may not have aged product available. And that there are several options. The first is we can use the representative extractable profile to focus the development. As I said, we typically use 3 solvents, one high polarity, one low polarity, and then one of them that simulates the product itself. We can use that to help guide that profile of what for the development. We could use the worst case scenario to look for making sure that we're doing optimization of the separations chromatographically, and then identify the extractables for the potential leachables from that subset. Another potential is to use simulation studies. Now, simulation studies are fairly well defined, and it's kind of, we would go in, fill a container closure system with either product and or placebo, sometimes both cases, and then artificially create leachables by putting them under accelerated conditions. A lot of times you can go in there 50, 60 degrees, significantly higher for several months, and that allows you to generate a potential worst case scenario from a simulation standpoint of what your potential leachables will be. Now, to jump in terms of elemental impurities, the first question is, from an elemental impurity standpoint, can we combine the two? And the short answer is yes, we can combine extractables testing with elemental impurities, because ICH Q3D and the USP do account for taking into account the effects from the container closure system. It's the only ICH guideline from an impurity standpoint that doesn't exclude leachables. So we can tie those in and use the levels, the tox levels that the ICH and USP refer to for our targets for our elemental analysis from an extractable standpoint. The second thing to understand is which applies to you, the USP or the ICH. Now, for the most part, they are harmonized between the two, but there are some minor differences. So it is important to understand when you're setting these up, which one applies to which. The first is the USP applies to those products which are monographed. So unless the monograph specifically states otherwise. So if you have a monograph material that's in the USP, then the USP chapter, i.e. 232, would apply. ICH applies to any new product that does not have a monograph. So any marketed products that were approved via an NDA or an ANDA and which do not have a monograph also must comply with the ICH. And all marketed products which were not approved via NDA or ANDA, for example, OTC over the counters, but again, that do not have a monograph, would need to comply with the ICH. And here you can see how the ICH relates and refers into the leachables in the USP of where to control those. So the elemental impurities here, again, are looking at leachables, so you need to evaluate your final container closure system and your final drug product. The other areas, again, are all the different sections of where elemental impurities and or leachables could come from. The utilities, that's your water, your manufacturing source, the equipment, the excipients, the drug substance. But again, here we're specifically looking at the leachable, so we're looking at what's coming out of the container closure system into our final drug product. So some challenges as we move into the leachables method development. We use some generic methods or screening methods to look at extractables. Sometimes those are going to comply directly to our leachables testing, other times it may not. Some of the challenges are that typically a lot of the leachable methods are more complex. You may need more complex sample preparations. Just because of either you need to look at mass spec or UV detection, or some of the leachables don't even have good chromophore, so you're going to be looking at chromophoric detection techniques, for example, ELSD, corona detection, mass spec. The sample preps can usually go through multiple levels of either derivatization or concentration, which makes it a little bit more challenging. You can use the extractables profile to help, of course, aid in development, but it's always important that they need to be developed and validated for your specific drug product using your specific dosing regimen. The other challenging part is that the quantitation limit, or the AET, is typically 10 to 100 times lower than the ICH impurity levels. So it's very common for the challenge here is we can see impurities when we're looking at leachables that are coming from the API or drug product, true degradants that you have never observed before. For example, we've been looking at drug products to where the client or the sponsor has says, well, we don't have any impurities. You don't have to worry about any impurity interferences from our product. It's very pure, it's very stable. But when you look at 100 times lower than that product has ever been evaluated, all of a sudden you do see that there are impurities. They're just not at the level that you would typically monitor for impurities at. This gets very challenging for biologicals, because a lot of times you would never look at a biological for impurities at that low. Now, we don't care about the drug substance or drug product impurities or degradants. The challenging part is they can interfere from a chromatographic standpoint and make monitoring for leachables much more complicated. And then you have to make a choice between an internal and external standard. There's two different techniques here where you're quantitating against an external standard. But then also internal standards can be very useful, especially for very complex systems or very complex sample preparations, that having an internal standard helps to correct for any potential biases that you can unfortunately run into with some complex, especially derivatization. If you have to do that, internal standards almost are a requirement at that point. Leachable methods, when you end up validating them, all the best practice guidances and the regulatory expectation is that they are validated as if any other regular impurity method, because they are monitoring that. So you have to walk through the normal ones. However, there are some caveats here. When you validate for accuracy, you may end up with accuracy wider than you would normally expect for an impurity. And this is really because a lot of times, as I kind of mentioned, you're at 100 times lower than what your normal impurity method is. You're dealing with a complex chromatographic system, so your acceptance criteria may need to be a little bit wider. Precision gets to be complicated from a homogeneity standpoint. For example, if you're dealing with MDIs, it's actually not uncommon to have leachables profile actually vary slightly from individual MDI to MDI. So when you're validating that, you need to, how am I going to make sure I'm looking at a homogeneous sample prep? And that usually means combining multiple different product or individual units together to make a homogeneous solution. And again, the acceptance criteria may be wider because you are dealing at a much lower level. Intermediate precision, the same issues that you have with precision. Linearity, you'll run from the AET to essentially 150% of your target specification. Typically in the AET, again, is the level at which basically it's your reporting level just for your leachables. Quantitation limit should be at or below your AET. Again, you're not going to worry about anything below the AET, so you want to make sure that you can see that. And specificity can get very challenging, especially with API formulation components at the levels that we end up looking at, where typically they may not be an issue from a specificity standpoint, but do become an issue just because of how low that we're going. And then, of course, solution and robustness testing are required as well. So stability study types. There are, ICH defines a variety of them. Long-term stability, this is the normal target storage of how you're going to store your product over time. For example, if your marketed or your hoped for storage is room temperature, this would be 2560. Intermediate, where the stability condition is designed to moderately increase the rate of degradation, so they're a little bit higher temp and humidity. Then, of course, accelerated, which is defined as the potential worst case predictive condition for long-term stabilities. Stress testing is defined by ICH, where you intentionally go in and you degrade them. It really does not apply to leachables because you're degrading your drug substance and or drug product. And then photo stability is where you're looking at the impact of light stability on your product. The main ones that we look at or worried about, of course, are the long-term and accelerated or intermediate stability conditions. Photo stability can come into play because if you have a container closure system that is photosensitive, you can get a different leachables profile if you end up with a photo-degrading container closure system. This is just the definition for general storage or refrigerated. Again, the long-term defined as for general storage or room temperature, 2560. The numbers I have there in parentheses and time is the length of data that is recommended by the ICH to be filed for registration. This is, again, from the guidance, it's only recommended. It's actually very common to submit with less data than this, typically no less than 6 months. You can actually file with 3 months, but then with a commitment that at the 3-month or 90-day update, you give the 6-month data. It's also important to understand that the agency will only evaluate essentially based upon for leachables the length of data that you provide. I know the ICH from a degradant standpoint allows you to extrapolate. Extrapolating for leachables is much more difficult, which I'll kind of cover here shortly of why that becomes much more of an issue. And then the other normal storage conditions, refrigerated, frozen, and semi-permeable. Excuse me. So leachable stability program essentially is going to be very similar to an ICH. 3 primary drug product batches on each drug product configuration. So if you have different dosing strengths, you need to evaluate each dosing strength. The caveat here is unless you set up a bracketing or matrixing design, and you can do that in some cases for leachables, if under certain circumstances you're using the same container closure system, exactly the same, and your formulation is similar. I would caution you about using bracketing and matrixing unless you completely understand it, because it can get complicated to justify the data, especially when you can end up with a lot of variance from a leachables profile where even going from, let's say, a 5 milligram to a 10 milligram concentration level can have a direct impact on your leachables profile in some cases. The batches should be all made by the same process of what you're filing with. That way you can ensure that you have the same material and that you're making all your decisions on. And then each batch recommended to be with a different batch of drug substance. That's just the ICH recommendation. From a leachable standpoint, it really doesn't impact it, but that's the recommendation from that. And the container closure should be the same as proposed for the marketing package, and it really almost is an absolute requirement. If you don't use the same container closure system, you can't necessarily use that data to justify a leachable stability program. You will also need to include the secondary packaging if it is needed. For example, if you have a laminated foil pouch as part of your secondary container, you will need to perform that on your stability at least for the leachables profile. So testing frequency, ICH and several of the other, from a leachable standpoint, recommends doing the same testing frequency as for impurities. First year every 3 months, second year 6 months, and then yearly after. Accelerated 3 times 0.03 and 6. And then intermediate, if you have an intermediate condition, every 3 months for 1 year. It is important to note that for leachables, sometimes it is acceptable to have an altered time frequency. Some places, if they've had data from previous batches, they may only test at 0, 12, and 24, or every 6 months. If you don't have a lot of data to back that up, I would not recommend that. It's also important to understand that you don't want to get surprised, go to your 12-month time point, and all of a sudden realize that you have a leachables issue that necessarily may not have been caught by extractables. One important thing that does come up is configuration. Leachables can be directly impacted by the storage configuration of your product. And it's pretty simple to envision why that can be by just looking at a typical meter dose inhaler. So if we look at this, the drugs is usually a suspension or a solution and an aluminum canister, but then it comes in contact with a gasket, metering valve, valve stem. And as you can imagine is if it's stored in this position as shown on the screen, the solution is coming in direct contact with the gasket, with the metering valve, so you can have direct interaction there. However, if it's stored other way, upside down, or as sometimes we refer to as valve up, then the solution may not come in direct contact with the gasket and valve, and all of it's coming in contact with the canister. The same can be true for example, vial stoppers solutions. So they stored stopper up, stopper down. I know normally it's all stopper up, but it's important to understand that how that configuration can have a direct impact on what the leachables profile is. So the orientation should represent all the possible different ones: upright, inverted, in some cases even sideways, depending upon what your product is. That needs to be evaluated on a case-by-case basis, and especially different multiple contact points. For example, it comes into contact with 2 materials if stored upright, 5 materials if stored, upside down. The storage should always include primary packaging with your labels. Inks and dyes are well-known sources of leachables. You may not get any leachables actually directly from the container closure system. For example, an HDPE bottle, but if you have a label on the exterior of that bottle, the inks and dyes have been known to migrate or leach directly through the bottle and then show up in your final product. So it's important to have them there. And as I kind of mentioned before, secondary packaging, it may be very important to ensure that you have the correct leachables profile, especially if you're dealing with a container closure system which is permeable, semi-permeable, LDPE, polymeric based typically. Those can be susceptible to leachables from a secondary packaging source. So when you get into, so you've gone through, you put everything up on stability, you've monitored them, now you want to track and trend and look at stability, because that's what you do in a stability program. Unfortunately, stability trending can be extremely tricky when you're looking at a leachables profile. The first is that if you look at what you observe, the Arrhenius equation won't apply, because you actually end up with a mix of kinetics that impact what's going on. The first are migration kinetics, and this is how fast will that specific leachable, can it migrate through the container closure system. So if you have an HDPE bottle, how fast can that leachable actually migrate through that physical polymer bottle over time? What's the rate of that? And that can vary significantly between different types of leachables. The next one is it can migrate through the bottle at a certain rate, but then it needs to be soluble or partitioned into either the head space, if there's a head space for example, in your bottle, or directly into your drug substance. So you could have things that can migrate very quickly through the bottle, but may not ever end up as a leachable because they're not soluble in your final container closures, or I'm sorry, your final drug product formulation. Or you can have the exact opposite, things that may migrate very slowly, but are going to be extremely soluble in your drug product formulation. So that's why sometimes stability trending gets to be tricky. Regression analysis can be used. Kinetic curves, however, can be somewhat different than what you're used to, because what you can run into, or what you will observe typically, is asymptotic leaching. So it's not like a degradant that necessarily is going to continue to be formed over time, because you're only starting off with a known amount. For example, in the bottle, you may only start off with 10 micrograms of that potential leachable. You can't physically leach any more than that out of there. So there's going to be a time where it starts to, essentially the leaching rate slows down and you reach a maximum. Now, accelerated additions can be more predictive, but they may not be more predictive. The challenge here is that, as I talked about with the migration kinetics and solubility, you can end up with cases to where temperature, when you have those two kinetics actually competing. So if you look at from a solubility standpoint, think about the polymer bottle as it has a solubility. There's a affinity between the leachable and the bottle, and you have an affinity between the leachable and your solution. And that varies with temperature. You increase that temperature, the solubility profiles for both will probably go up, but probably not at the same rate. So you can actually have a case where at increased temperatures, you could or can have lower leachables because they're going to have a greater affinity for the bottle than the solution than they do at room temperature. Here's an example of an asymptotic leaching profile. As I kind of mentioned before, this is a case where only 10 micrograms comes out. So over the time period, if you were to track and trend and try to extrapolate a expiry, or when you're going to hit a certain level, and you're using that as 6 months data, well, then you're going to end up with a much shorter possible expiry than looking at a 12-month data point. Because as you can see, if you were to draw a extrapolation curve at that 6-month time point, which is approximately at 8 micrograms, it would say that by, essentially almost by 15 months, you would have, 20, 25 micrograms there. But it maxes out at 10 because that's all that was there. So this gets where it gets a little bit more tricky from a trending standpoint, and extrapolating is also why the FDA typically will say, we want to see real world data of what your maximum leachable profile is, be able to set those specifications from a tox assessment standpoint. So you're performing this leachable stability. Hopefully you've identified your leachables before you go into your registration stability batches, but it is potential you may not. So any leachable that you do observe above the analytical threshold needs to be identified, and at minimum a paper tox assessment, QSAR analysis needs to be performed to evaluate what its toxicological concern level is. Is it a compound that needs to be concerned about or not? And this tox evaluation can be significantly different than what you do for normal impurities, and that's just really because of the different types of compounds that you may have as leachables. All your drug product impurities are likely going to be related to your drug substance in some form or fashion, so it may have higher or lower toxicity, but it may not be significantly that different from it. Leachables, you're dealing with a wide range of compounds, from elastomeric components to colorants to plasticizers, non-slip agents, and a wide range from dealing from very polar, very small molecules to very big polymer fragments. So you can end up with a wide range of toxicity concerns. So it's important to make sure that anything above that AET, you do the proper overall tox evaluation on it. The end goal in a lot of this is try to establish an extractable leachable correlation. And what that allows you to do is predict in the final leachable how much of that leachable is going to be present. So the idea is that if you've appropriately qualified or characterized your container closure system, you can say that, for example, that I give here, that you have 30 micrograms of that extractable. Let's say it's compound A. So there's 30 micrograms of compound A in your container closure system. If you can establish extractable leachable correlation with data, then you can definitively say that that's going to correlate to 2 micrograms in the leachable. What that allows you to do is that if you can make that correlation, from a regulatory standpoint, what that can allow you to do is not to have to do full-blown leachable testing on your final commercial product. Because if you can establish that extractable leachable level, you can put a control process into place that maybe you're going to control it coming into your container closure system by performing extractables testing on your bottles, and your specification on the bottle may be 30 micrograms because you know that 30 micrograms will only relate to 2 micrograms in your leachable. But you have to get data to be able to do that correlation. Now, there's a number of factors which will influence in leaching. Some of these are similar to, of course, drug product, what's going to impact the degradation profile. Some of them are not. It's important to understand how some of these can have an impact on your leachables, especially if you're looking at life cycle concern or changes to your process. Of course, temperature and solvents are the easiest things. Ionic strength and pH can have a very significant impact. very small changes in pH or ionic strength can have a dramatic impact on the leachable, especially for some leachable components. Chelating agents have been known to allow for leachables in your final product, which typically wouldn't be there. Again, I kind of already covered the drug product characteristics. Salvation aids. In today's saying, a very big number or a very large number of drug products now are semi-soluble or very low solubility products. So it's very common to add in solubility aids, Captazole or other types of solubility aids to help those compounds go into solution. They do that great. They allow for non-soluble or low solubility compounds to go into solution in aqueous systems. They also allow leachables to go into the final product that typically wouldn't leach. So they do both very well. Excipients can have it. Packaging stability, i.e., is your packaging going to be stable, or as it breaks down, is it going to form new leachables? And irradiation or sterilization. So if you sterile product, the means by which you sterilize it can have a direct impact on your leachables profile. So it's important to do all of these studies on materials that have already been sterilized because it will impact your leachables profile. So some very common questions that do come up. If I don't have any extractables above the AET, am I done? I wish that was the case always. The short answer is it depends. And sometimes it depends upon going back to that risk assessment table. If you have a solid oral or a dermal product, then the short answer is probably yes. If you don't have any AETs, or I'm sorry, any leachables or extractables above the AET, you could probably say yes, I'm done. If you're dealing with a parenteral or especially an inhalation product, then you're going to need to end up generating leachables data to back up that nothing is leaching that you didn't observe during your extractables. What if I can't monitor down to the AET level for leachables? And this is actually a common issue, especially for larger volume parenterals and some dosage forms, to where when you calculate the AET or level you have to look down to, you just physically can't go down that low, because sometimes these levels really start to push the technology that we have. Now you can get there with going by some extreme measures of concentration, going into mass spec levels, but it can still get very difficult to push these levels down. In those cases, what we typically recommend is to go back and characterize the extractables. Go up a level, identify all of your extractable peaks that do correlate down to it, and use that as your worst case scenario. And we've seen that be successful in multiple times. Will it be required to have routine leachables testing? Part of that is going to depend upon, of course, your extractables and what leachables information you can present. If you can present a leachables profile that you can demonstrate you don't have leachables, then of course the short answer is no, you probably will not have to have routine leachables testing. However, if you're a MDI, a meter dosed inhaler, pressurized, it's almost guaranteed you're going to have to do routine leachables testing. The vast majority of parenteral products end up with requiring routine testing, and then the others are much more hit and miss. Of course, solid orals routinely do not require any routine leachables testing. What if I can't identify all the compounds above the AET? This can happen. Sometimes you may have a leachable that you can't isolate because as you attempt to isolate it becomes transitory. Perhaps it's intermediate, it degrades itself. Maybe a leachable that isn't able to get mass spec because it's not ionizable or something. If you do your due diligence, what the agency typically looks for is if you have 10 leachables, you've identified 9 of them, and you can demonstrate that, yes, I've done due diligence of trying to identify it. We've seen that to be successful in terms of the regulatory approach. You can assume or make the assumption that that last leachable will have a similar toxicological impact as the worst case one that you have identified. The other potential is it may not be able to identify the specific compound, but if you can at least identify the class of compound of that leachable, that'll help out significantly. And now just wrapping up, a couple of quick case studies. The first is a lyophilized product. It is a parenteral, 20 millimeter vial with a bromobutyl stopper. Aged product was available, stored in the final container closure system from a phase 2 study. So the path forward here, of course, was we did a controlled extraction study on the stopper. Using the aged product that was available, correlated what we saw from the controlled extraction to what we saw in the aged product, developed the Leachables method, and then tested all together again from the aged product along with the extractables profile samples. No leachables were observed in the aged product above that AET. And so what we were able to do is that since it was a lyophilized product, it kind of falls into a medium risk association. We were able to basically make the good regulatory justification that no formal stability studies were required, and this product was accepted and actually was approved by the agency. A second case study, this is a pressurized meter dose inhaler. A full extractable leachable study was performed, as is typically required for almost all extractable leachables on a MDI, meter dose inhaler device. A new unknown was detected at 12 months, and when we looked at that, it did not correlate to anything that was observed in an extractables profile. However, when we identified it, it was related directly to a known polymer additive from the structural relationship. When we went back and did some additional extractable source, we were able to confirm it, but it was a case where we had to do extreme extractables to extracting significantly more material than you would typically ever do. So this is kind of one of those cases of we found a leachable, but it didn't necessarily originally go back to the extractable. We were only able to find it when we went to beyond extreme extraction conditions to be able to confirm, yes, it actually was coming from part of the container closure system. And it was that unique environment of the MDI that was essentially allowing it to leach when we didn't observe it previously under normal extraction conditions.